So it's time to start this joint webinar between the International Eosinophil Society and European Mast Cell and Basophil Research Network, EMBRN, on the topic from infection and stress to allergy, roles of eosinophils and mast cells. So my name is Gunnar Nilsson. I'm the president of EMBRN. I will co-chair this joint webinar together with Allison, who is the president of the IES. So first, a few words about the European Mast Cell and Basophil Research Network, the EMBRN, which is then, although it's called European, it's a global network for all those who are interested in the biology of mast cells and basophils and the diseases associated with these cells. EMBRN started in 2004, and in the beginning we had annual meetings. Nowadays we have biannual meetings, and the next meeting will take place in June in Vienna, Austria. You can see on this slide that we have some confirmed speakers, and the local organizer, it's Philipp Starkl, that you will hear from later on during this webinar. And he's working very hard to put together a fabulous meeting that we invite you all to. If you're EMBRN member or IES members, you're all welcome. So please, Allison.
Thank you so much, Gunnar. It is absolutely, I'm delighted to be here today. So I am the president of the International Eosinophil Society, which is an organization of scientists and clinicians who are interested in, of course, the eosinophil and all the diseases that cell is involved in. We also sponsor biennial meetings, but also monthly webinars, of which this is one, and other educational programming for our over 300 members to review all sorts of information about the eosinophil and share our research. If I can have the next slide, thank you. I wanted to let you know about a really exciting opportunity. The Eosinophil Society and EMBRN are collaborating so that if you join one of the societies, you can get free membership or complimentary membership in the other. And so on this slide, you can see two little QR codes at the bottom, and you can choose to join either the IES or EMBRN, and then from there, you can link to the other society as well. I do want to let you know that membership in the International Eosinophil Society is free and is open to all pre-doctoral individuals, as well as those who are in postdoctoral fellowships. So we offer complimentary membership to all trainees at this point in time. Our next conference is going to be in 2027, and it's going to be in Vancouver, Canada. So please mark your calendars. I know it's planning a long way in advance, but we meet every 2 years, and we have just begun putting together our scientific program for this meeting. So it's going to be extremely exciting, and please come join us in Canada in another year and a half. If I can have the next slide. The next webinar that we will be hosting will be about prenatal and early life shaping of eosinophils. We have 3 fabulous speakers lined up: Dr. Joan Cook-Mills, Dr. Jen-Ying Nai, and Dr. Ian Lukowicz. So please mark your calendars. That webinar will be in February 11th. I would also like to thank our Corporate Advisory Council members. So we have AstraZeneca, GlaxoSmithKline, Amgen, Sanofi Regeneron, Celldex, Takeda, and Uniquity Bio. And all of these companies help to contribute to the running of the Eosinophil Society and to supporting these webinars. So thank you very, very much. So a little bit of housekeeping as far as today goes. Dr. Nilsson will be introducing the 3 speakers. You should have a question and answer button at the bottom of your screen. It should look like a square with a little question in it. Please use that button to type in any questions. You can ask questions at any time during the presentations, and then Dr. Gunnar will help moderate the questions at the end of each speaker's session. All attendees should be muted, and the chat feature is disabled, so please don't use the chat feature. The webinar will be recorded, and then we will post it onto our IES website for your viewing pleasure at some point in the future. And if you want to use closed captioning or live transcription, you can click the show captions button at the bottom of your screen. So with that, I'm going to turn it over to Dr. Nilsson to introduce our 3 speakers today. Thank you so much.
Dr. Nilsson, you're muted.
Thank you, Kira. So we have a great lineup for today's webinar. It's Dr. Philipp Starkl from Vienna and Nicolas Gaudenzio from Toulouse and Jean Marshall from Halifax. So we will start with Philipp Starkl, who is the principal investigator at the Medical University of Vienna. He earned his PhD in immunology from this university, and then he completed a postdoctoral training in Steve Galli's laboratory at Stanford University, where he discovered a novel protective role for IgE antibodies and mast cells in adaptive immunity against insect and reptile venoms. He later returned to Austria as a senior postdoc at the Medical University of Vienna, and since 2023, he has been an independent group leader at the Medical University of Vienna, focusing on how microbial infections interact with type 2 immune responses in barrier organs and shape long-term immune outcomes. So today, Philipp will talk about eosinophil, innate immune memory after bacterial skin infection, promotes allergic lung inflammation. Please, Philipp.
Hello, everyone. I will try to share my screen. I hope you can see my full screen now.
Yes.
Okay, great. Yeah, okay, then let's take it away. Thank you very much, Gunnar, for the kind introduction. And I would like to thank the organizers, and especially the IES, for the invitation to speak here at the webinar. This is really a great opportunity. And as Gunnar already indicated, I have to disclaim that I'm actually originally coming from the mast cell field, but we have recently tapped into the fascinating world of eosinophil biology. And in my talk today, I will try to give you a concise run through one of our most recent studies where we discovered that skin infection can actually lead to long-term reprogramming of eosinophils and that this can have consequences for the development of allergies. I'm sure many of you are familiar with this equilibrium model of our immune system. Here, the different opposing arms of immunity are assigned to specific host defense functions. We have type 1 immunity against intracellular microbes. We have type 2 immunity that is especially efficient against parasites. Type 3 immune responses against extracellular microorganisms like bacteria and fungi. And then we finally have a type 4 response that is kind of a universal preventative strategy mediated predominantly by secretory IgA antibodies. I think that this is a nice way to explain the different modules and specialities of our immune system. But on the other hand, I believe that the reality is more complex with rather blurry boundaries between the different type 1, 2, and 3 responses. For instance, we know today that not only parasites, but also viruses and bacteria can trigger type 2 immunity. One example for such a bacterium with close association with type 2 immunity is Staphylococcus aureus. Staph aureus is really a remarkable pathogen, and it's a common and frequent colonizer of the skin and mucosal areas. Staph aureus can produce different toxins that help it to defend its niche in the microbiota, but when the opportunity arises, these toxins can help it also to invade the host. And the infections caused by Staph aureus are an important health problem, as many strains are antibiotics resistant and there are still no effective vaccines. One strong indication that Staph aureus can trigger type 2 immunity comes from human allergic patients where atopic dermatitis and asthma severity correlate quite strongly with levels of Staph aureus toxin-specific IgE antibodies. Since we found this quite fascinating and we have a long-standing interest in type 2 immunity and infectious diseases, we wanted to have a closer look at the immune response to Staph aureus and what it means for systemic immune responses. So this was the starting point of our study that was led by Mariam Radouani, a fantastic former PhD student in the lab who is now a postdoc in Bart Lambrecht's group at the VRB in Ghent. Since Staph is a frequent colonizer of the skin, we decided to use a model where the animals are first shaved and then tape stripped and exposed to a patch inoculated with Staph aureus or PBS for 1 week. It is important to know that this epicutaneous infection model, the bacteria are restricted to the skin and don't cause severe disease by systemic spread. Using this setup, Mariam first analyzed the molecular and cellular skin immune response. Apart from the kind of expected antibacterial type 3 response that is reflected by increased levels of IL-17, but also IL-22, it was very interesting to see that also the levels of pro-inflammatory molecules with important roles in type 2 responses, like the alarmin IL-33, but also IL-4 were significantly increased. Among the analyzed immune cells, we found a quite fast and progressive infiltration of neutrophils and eosinophils upon Staph infection. This was a quite clear indication for us that the Staph-induced skin inflammation was strong enough to mobilize cells in the bone marrow. Even though especially the neutrophil infiltration at the site of inflammation is not really surprising, we found the idea that the inflammation caused by this non-invasive skin infection reaching and affecting the bone marrow compartment pretty fascinating. So we then had a closer look at the bone marrow, and we found that among all the analyzed mature immune cells and their progenitors, only the eosinophils and the eosinophil progenitors were significantly expanded on day 7 after the infection. We next performed single-cell RNA sequencing of whole bone marrow, and among the identified cell populations, we observed the most profound effects among the eosinophil and eosinophil progenitor compartment. As we could not faithfully identify and assign the eosinophil populations in our 10x results, we specifically sorted bone marrow eosinophils and their progenitors and performed bulk RNA sequencing. In these experiments, we found that the skin infection led to differentially expressed genes in these populations that related to various physiologic functions such as cytokine signaling, metabolism, or reactive oxygen species production and responses. So these data indicate that the localized non-invasive Staph infection had significant effects on the transcriptome of bone marrow, mature eosinophils, and their progenitors. The observation that epicutaneous Staph infection can trigger features of type 2 immunity and the skin, and that it affects eosinophil infiltration made us wonder whether such skin environment could pave the way for allergy development. Mariam tested this by extending the mouse model to a sensitization phase with 1 week epicutaneous exposure to house dust mite extract. To investigate systemic effects, we then challenged the animals with a single intranasal house dust mite application on day 14. On the next day, it was fascinating to see that the mice with the prior infection responded to the intranasal allergen exposure with dramatically higher lung cytokine production. These increased inflammatory cytokine levels were also accompanied by substantially higher neutrophil and eosinophil levels in the lungs. To better understand the consequences for more established allergic airway inflammation, Mariam further extended the model to house dust mite treatment intranasally for 1 week. Also in this setting, the effects on the increased eosinophil numbers were still present. In addition, we also saw that the mice with the prior infection showed profoundly increased allergen-specific IgE and IgG1 levels, as well as impaired lung function. We think that these results show that local Staph aureus skin infection can indeed be an efficient primer for dermal allergic sensitization and ultimately systemically increases susceptibility to develop allergies. But what could be the mechanism behind this increased allergic susceptibility and and eosinophil mobilization. First, we wanted to investigate the importance of the effects on the bone marrow. For this, Mariam established a bone marrow transfer setting where she first infected ubiquitously GFP transgenic mice with bacteria with PBS and collected the bone marrow on day 7. She then transferred the bone marrow into irradiated GFP negative wild-type recipients, and after 5 weeks of reconstitution, we continued with the second part of the model and exposed the mice to hostosmide extract on the skin and the single intranasal allergen application. I have to say it was probably one of the most striking moments of the project for me when we observed that even after 5 weeks and in mice that have never been directly exposed to the bacterium, the increased eosinophil infiltration in the lungs in response to the allergen treatment was still detectable. We did not observe such an effect in neutrophils, and there were no quantitative difference in the blood or bone marrow eosinophils, indicating that the effect was not just simply caused by more efficient cell engraftment. We rather suspected that it was a consequence of long-lasting qualitative modification of bone marrow progenitors triggered by the skin inflammation. To test this idea, we differentiated bone marrow of control or in infected mice ex vivo into eosinophils using a protocol that lasted 14 days. While the infection did not affect the purity of our cultures, the eosinophils differentiated from mice with prior infection were substantially different. For one, we found significantly altered expression of various surface molecules. But more importantly, the cells differentiated from the bone marrow of mice with skin infection showed quite striking functional differences, with increased production of cytokines and increased release of granules peroxidase after exposure to hostosmide extract. The eosinophils differentiated from the infected mice also showed profoundly different metabolite content that indicated reprogramming related to protein synthesis, aerobic glycolysis, and the pentose phosphate pathway. We then also analyzed the global epigenetic state by attack sequencing and found quite unexpected results. The infection was associated with many more closed chromatin regions. Many of the regions were associated with genes related to protein synthesis and general housekeeping functions. On the other hand, the gene regions related to more specific cellular functions like motility, host defense, and granule formation were more accessible, indicating a higher potential for transcriptional activity. We interpret these results as an indication that the infection-induced priming of progenitors results in differentiated cells with clearer defined cellular functions that are more efficient in causing inflammation. Overall, we conclude from these experiments that the skin infection influences independent pulmonary inflammation by reprogramming of bone marrow progenitors. This inflammatory imprinting of progenitors results in fundamentally reconfigured eosinophils with altered receptor expression, function, metabolism, and epigenetic setup. Finally, we wondered about the potential mechanisms that could drive the allergic lung phenotype. So we revisited the inflammatory patterns in the different tissues throughout our model, looking at the skin cytokines after priming on day 7, after allergen sensitization by the skin at day 14, as well as in the lung on day 15 after the intranasal hostosmide allergen exposure. We realized that there were 2 mediators that were consistently increased, the alarmin R33 and the complement anaphylatoxin C5a. Further underlining a potential role of these mediators, we detected enhanced expression of the C5a receptor as well as ST2, the receptor for R33, by bone marrow eosinophils in mice with Staph aureus skin infection. This evidence prompted us to investigate the importance of C5a and the R33 receptor axis. To do this, we generated mice with eosinophil-specific receptor knockouts using Jamie Lee's EOCRE mice. First, we combined the EOCRE strain with Jörg Köhl's C5a receptor flox mice to test the importance of C5a signaling. In control animals, we actually found the same phenotype as before in normal wild-type animals with increased eosinophil infiltration into the lungs after allergen challenge. It was quite striking that the animals with eosinophil-specific C5a receptor knockout showed actually a blocked infiltration of eosinophils into the lungs when C5a receptor was specifically absent from the eosinophils. In contrast, the numbers on the bone marrow eosinophils and the eosinophil progenitors were not affected by the eosinophil C5a receptor deficiency. We then used a similar strategy to specifically knock out the IL-33 receptor ST2 in eosinophils. In the lungs, we observed a quite similar phenotype as before with the C5a receptor knockouts, with significantly lower pulmonary eosinophil counts. But this time, also the expansion of mature eosinophils and the progenitors in the bone marrow was blocked, indicating that IL-33 signaling was quite important for the observed bone marrow effects. The final, but I think very important piece of data that I would like to share with you today is this one. No matter based on which strategy the eosinophils were absent from the lungs, either by the C5a receptor or the ST2 receptor deficiency, together with their absence, the inflammation in the lung was substantially diminished. And I think this is critical data as it shows the immediate importance of the reprogrammed eosinophils for the increased allergic lung inflammation in our model. Overall, I think we are looking at a remarkable mechanism of eosinophil-mediated inflammation that depends on two key mediator axes. While IL-33 seems to be critical for the effects on the bone marrow, C5a seems to be the major driving force of eosinophil recruitment to the lungs. I would like to end here with a graphical model that summarizes our conclusions. We think that local skin infection can pave the way for increased cutaneous allergic sensitization with consequences for allergic airway inflammation and asthma. This is reflected by increased pulmonary cytokines, IgE antibodies, and overall lung inflammation. We think that one of the major mechanistic routes involves local production of IL-33 in the skin, leading to an expansion of eosinophils and the progenitors in the bone marrow. The infection leads to long-lasting reprogramming of eosinophils that show different features of innate immune memory. And the eosinophil imprinting seems to be an important reason for the increased allergic lung inflammation that appears to be critically dependent on more efficient eosinophil recruitment via C5a, maybe also via IL-33. With this, I would like to thank my great lab and our friends and collaborators who contributed to this work, and of course our sponsors. Special thanks and respect go again to the fantastic and brave Mariam Radouani, who spearheaded this study. Now, with my final slide, I would like to cordially invite you to the upcoming EMBRN conference, which we are very excited to host this year in June in Vienna. I'm really stoked about our preliminary program with a lot of fantastic speakers, and I think this will be really an exciting conference. And spoiler alert, there will also be a session on eosinophils that is jointly organized with the International Eosinophil Society. So if you're a type 2 granulocyte fan, I hope to see you in Vienna in June. I think it's going to be a great meeting. And we expect to open our registrations and the abstract submission in February. So please follow us on social media to stay up to date. So now I'm really at the end, and I would like to thank again the organizers and the IES for the invitation to speak. And of course, I would like to thank all of you following online for your attention. Thank you very much.
Thank you so much, Philipp, for a fascinating presentation. Nice work that you have done.
Thank you.
So please use the Q&A for your questions. And we have a couple of questions already. So from Kelly McNagny, he asked about, excellent presentation. What do you make of the dramatic drop in type 3 cytokines in the C5a receptor and ST2 knockouts? The IL-22 and IL-17, etc.
Yeah, thank you very much, Kelly. This is a great question, and we have wondered about this ourselves. So I think there are two explanations for for this observation on the one hand could be the eosinophils directly that produce these cytokines. But to be honest, this is something that we have not looked into it, and otherwise it could also be an indirect effect on the compounds produced and released by the eosinophils, and that it's kind of a response to the degranulation of the eosinophils on site. But it's an excellent question, but unfortunately we don't have an answer yet.
Judah Denberg writes, fascinating and excellent talk. Any observation on eosinophil progenitors in the skin?
Thank you, Judah. Interesting and very important question as well. We only looked for eosinophil progenitors in the bone marrow. We did not look in the skin. But so that being said, can't exclude that they are there as well. Very interesting, and we should look at this.
And from Jenny Hallgren, Martinsson, can you see more IL-33 in the bone marrow after a skin infection? If so, do you think this is due to IL-33 produced in the skin or other mediators in the skin that induces IL-33 in the bone marrow?
Mm-hmm. Hi, Jenny. Thanks for the great question. We tried to pin down the source of IL-33 in our model using reporter animals, and we did not observe any cells in the bone marrow that produce it, but we saw a source of IL-33, at least an increased IL-33 signal in the skin, in the fibroblast compartment in response to the skin infection. We also tried to detect IL-33 in the bone marrow using an ELISA setup, but there we did not detect any. So our current view or hypothesis is that the IL-33 is produced in the skin and that it's a skin-derived IL-33 causing this effect.
And then we have from one anonymous. In your study, did you examine T-cell responses during primary staph A infection, as well as following sensitization and challenge with HDME? If so, were there any differences in the magnitude or phenotype of the T-cell responses between these conditions?
Yeah, thank you very much. We actually did quite some characterization of the T-cell responses in our study, and the data all contained in our manuscript. So I invite you to check it out, because I might be actually not be able to recapitulate everything 100% correct. But what I think to remember is that the skin infection increased the T follicular helper responses in the draining lymph nodes of the skin. So also against the house's mite extract, and overall we saw that the T-cell responses were enhanced in mice that had been previously treated with the skin infection. And also something that we observed but did not publish is that it requires really the second step of skin sensitization to house's mite extract to be able to see this increased allergen-induced lung infiltration of the eosinophils. Maybe this is also an interesting detail. So there is an adaptive component in this phenotype that is playing an important role, but we are not yet sure how this exactly works.
And then from Mark Rutenberg, do you suspect that the upstream events triggered by stuff that mediate the eosinophil memory are directly related to TLR or secondary to the local infection triggering the response?
Hi, Mark. Thanks for this important question. I personally think that it's several different factors that are contributing here. I think on the one hand, it's the inflammatory factors that are produced by the host in response to the infection. But on the other hand, there are also key roles of the Staph aureus toxins that cause that causes the skin inflammation that are important to drive the inflammation. And we have not yet detected any circulating Staph aureus toxins or bacterial component in our model. But I would also assume that while we don't see a systemic infection, that many of the Staph aureus toxins and soluble compounds that are produced or derived from the bacterium can actually reach the bone marrow. So I don't have a definitive answer at this point, but I would personally believe that it's a mixture of direct cellular stimulation by staph toxins or also microbial patterns and inflammatory mediators that are produced by the host in response to the infection.
We have a similar question from Sabine Stigeman-Konechski. She also asked which strain you used in the study, if you remember. Otherwise, you can check it up in the.
No, I remember that strain. We have used it quite a bit. Thank you, Sabine.
Yeah.
We used the strain USA 300, which is kind of a clinical isolate. So it was originally isolated from a patient, and it's very widely used. So the downside is of this strain that it's not a natural colonizer of mice. So we needed to use an epicutaneous infection model where we exposed the mice on the back skin with a patch of Staph aureus. So this is maybe a disadvantage because it doesn't reflect the natural Staph aureus colonization of the mice. But on the other hand, for us, it also has a great advantage that the mice then can efficiently clear the infection very efficiently. So after 1 week, we are unable to detect any more bacteria in the skin, and this allows us to actually investigate the setting of prior infection rather than chronic colonization and inflammation that might be caused otherwise.
And then we have two questions, one from Mark Benamo and one from Amali Samarishinge about how this relates to humans and human allergic patients.
Yeah, thank you both for this important question. I'm thinking about this a lot myself, I have to say, and I think a lot in the field of trained immunity, innate immune memory. I think that we still have to validate to which extent this is really translatable, because we are investigating here a setting of an experimental setting where basically our mice have never been exposed to such an inflammatory trigger before, while we as humans throughout our life, we are continuously and frequently exposed to infections and inflammation. So the question is to which extent a similar infection in a human could actually, let's say, still prime our already well primed and matured immune system as adults. And I think this is an important question. We have not looked at these kind of things in humans, but it will be very important to actually investigate this, also specifically in the context of staph infection. And we hope we will be able to do this.
And the last question from myself about the C5a receptor. Is it expressed on the bone marrow eosinophils and the eosinophil progenitors?
As far as I remember, we found it on the eosinophil progenitors and for sure also on the mature eosinophils.
In the bone marrow?
Yes, in the bone marrow.
Mm-hmm. There are a few more questions on the QA, and maybe you can answer those later on, Philipp.
Yeah, we'll be happy to do so.
So thank you all for all good questions, and thank you, Philipp, for your presentation.
Thank you very much, Gunnar. Thank you, everyone, for listening and for the great questions.
So we move on to the next speaker, who is Nicolas Gaudenzio from INSARM, where he's a research director and also chief scientific officer at Genoskin in Toulouse. He earned his PhD in immunology from the University of Toulouse and then completed a postdoctoral training at Stanford University, also in the laboratory of Stig Alley, and I guess you were there at the same time as Philipp, at least partly. You overlapped. And during your postdoc period, you started to study neuroimmune interactions and also examining how nociceptors can regulate immune responses. particularly in allergic and type 2 immune reactions, like those seen in atopic dermatitis. Your research now in Toulouse has continued this focus on neuroimmune interactions in health and disease, and we will hear a very fascinating story that you have published on maternal stress triggers early life eczema through fetal mast cell programming. So please, Nico.
Thank you, Gunnar. Thank you very much for the kind invitation. It's really a pleasure to be here. I'm going to share my screen. All right, you should be able to see it. Okay.
Looks perfect.
Okay, excellent. So this is my disclosure. And so as Gunnar mentioned, my lab focuses on how sensory neuron and immune cells actually interact together to regulate each other and how they regulate chronic inflammation. So we focus on atopic dermatitis, on a lot on mast cell ontogeny and biology, more recently in contact dermatitis and on the interface between the mother and the fetus and how this prime to pediatric disorder. This is the story that I'm going to tell today. And we also investigate this in patients and on ex vivo biostabilized human skin from Genoskin. So I'm not going to really introduce mast cell to the audience, but they are exclusively tissues and immune cells. They interact with blood vessels in the tissue, and we published in 2019 that mast cell in the skin that express the receptor MRGPRX2 in human or MRGPRB2 in the mouse, which is the receptor for substance P in mast cell. They form what we name sensory cluster with peripheral sensory neuron in the skin, and those clusters are capable of detecting allergens. And this interaction between substance P producing neuron and MRGPRX2 mast cell, what the kickstart of the development of type 2 immune response and features of atopic dermatitis. Then we perform intravital imaging, and then when you selectively engage sensory neuron, you will see that the mast cell nearby start to degranulate. So we propose that MRGPRX2 could be a new therapeutic target to treat mast cell-dependent inflammatory skin disorder, in particular atopic dermatitis. The very same year, 3 other articles were published that shows that this cognate interaction between mast cell and sensory neuron regulate much more aspects of the skin than type 2 inflammation, but also pain, also non-histaminergic itch. And this one is in collaboration with our lab as well, antibacterial response via direct sensing of bacterial product through MRGPRX2. This is coming from the labs of Ching Song Dong and John Hopkins and Brian King at the Mount Sinai. So we next really went to know more about the ontogeny of mast cell, what was the phenotype of those, MRGPRB2 positive mast cell, and we found that those ones that seed basically the skin, they come from the primitive hematopoiesis and especially the yolk sac. They will have a specific gene signature, notably enriched in neuropeptide receptor. They will seed virtually all tissues, and in any tissue that you can look at, they will be always found in contact with sensory neurons. On the other side, you have mast cells that are more the one that we recently call usually mucosal mast cell. They do not express MRGPRB2. We found that they actually do not develop from primitive hematopoiesis, but they develop after birth. You will need microbiota-derived signal for them to fully develop, notably in the gut. We use a fate mapping system in collaboration with Florent Ginoux in Paris, and found that they are actually not coming from the GMP state, as it was previously mentioned in textbook, but they might be coming from an intermediate stage between CMP and GMP. So this is still need to be identified correctly. They will have a completely different curve signature. And as you know, they will be mostly found in the gut, a little bit in the stomach, and a little bit in the lungs as well. There is this dichotomy that exists in the mouse. This is under homeostatic condition. If you trigger type 2 inflammation, the group of Kenji Kabashima nicely show that you can have the recruitment of bone marrow-derived cells that will then adopt in the skin a phenotype of MRGPRB2 positive connective tissue type mast cells as well. So the two compartments are kind of interconnected during inflammatory condition. When you switch to human, it's completely different. What we found is that we couldn't identify any CTMC, MMC dichotomy in human. We found that there is a steady state in different organs, 6 different, you can call them, state of mast cell. We don't know if they are actually, different subset or population. So we identify them as clusters. Among which the cluster number 3 correlated with what was previously described by Dan Dwyer and Josh Boyce actually in nasal polyps as well. So there is much more work to be done actually in human to better understand the heterogeneity of mast cell and how they react. And we wonder whether actually the mouse model is very good as we don't find this dichotomy in the humans. So we are pushing in collaboration with Laurent Robert to identify some tools to really study the reaction of human mast cells and notably in terms of drug development, because it's really important as, they will not necessarily share the same receptor than the mice. So we recently published a characterization of 2 humanized mice models in which, CD34 positive bone marrow progenitor from human are injected, and they develop some human immune system within the mouse background. And we found that the NSG SGM IL-15 recombinant humanized model is actually one of the best one if you want to have nice population of human mast cell for efficiency of drug engagement in vivo. To pursue just a small teaser that is still unpublished in collaboration with Laurent as well. We develop an FC enhanced actually anti-human KIT antibody, and we use that to wipe out mast cell very efficiently. And we used the biostabilized natural human skin model from Genoskin that you can basically inject like you would inject a patient with those antibodies, and in 7 days, basically you can create a mast cell deficient model that efficiently deplete human mast cells. So we are now combining humanized mouse models, natural human skin, preserved alive ex vivo to try to investigate, human skin mast cell behavior and drug targeting of those human skin mast cells. So now I'm going to switch to the part of the study related to prenatal stress, and notably the relationship between prenatal stress and what we name pediatric eczema, or sometimes is also named pediatric atopic dermatitis, but we think it's actually a different disorder. So as you know, 20% of infants develop pediatric eczema in the first 4 years of life. It's characterized by tissue inflammation, increased mechanical sensory sensitivity, which can worsen itch, disrupt sleep, impact quality of life. So there is an inflammatory component, but there is also a neurosensory component of the disorder. In this very nice graph from Thomas Bieber, basically that relates the onset of and the severity of the pathology depending on the age. We were very interested in this very early onset of atopic dermatitis, with remission. So those kids, they would develop flares and eczema within the first days after birth. And then for 70% of them, they will go in full remission of the pathology around the age of 5, 6. So we thought, this is too early for getting fully sensitized to develop feature of type 2 inflammation. And there were some Scandinavian studies that shows that maternal exposure to stress would eventually increase the risk of eczema in kids. So we wonder whether environmental perturbation in utero would predispose to pediatric eczema at birth. So to study that, we collaborate with the lab of Nicolas in Toulouse in France, to develop a model of prenatal stress. So pregnant downs are put under bright light 3 times a day for 30 minutes each time for 6 consecutive days. And then after that, we analyze the offspring, at, for example, week 8 and between week 0 and week 8 after birth, and see what's going on. So what we found is that those pups after birth, they have a normal skin. It seems that they do not develop actually what features of pediatric eczema with a normal distribution and percentage of immune cell at birth. However, there is something that we did not take in consideration is the fact that pediatric eczema occurs at fluxural location in the skin, often exposed to mechanical friction, such as you can see it here. So we wanted to mimic this friction in humid environments. It's the case, for example, for a diaper. So what we did is that we shaved the mice and we placed just a PBS filled gauze pad occluded with a dressing, on the top of the mouse. Such a mouse, when the mouse is walking, the gauze pad is slightly, performing a light, wet mechanical friction on the back. So if you do that on pups that are born from a healthy mother and controlled pregnancy, nothing happens. They have a completely normal skin. However, if you patch PBS just on the pups that are coming from a stressed mother, they start to develop a very severe skin inflammation that share features with eczematous lesions. And what was very interesting for us is that exactly like in the clinic, if you let the mouse grow older, then the phenotype is naturally lost with the age. You can see 6 months old mice, you cannot trigger this light, wet friction developing eczema actually in the skin. So we said we might have actually a model that mimics some of the features that are observed in the clinic. So we wanted to dig a little bit into it. So first of all, to analyze, the sensory component of the disease. So we analyze sensory neuron in the skin and their branching. In green, you have a staining with beta tubulin that stain most of sensory neuron projection that are coming from the dorsal root ganglia. And then in red, you have tyrosine hydroxylase that will stain for what we name silo threshold mechanoreceptor, but also sympathetic fibers in the skin. This is how, the staining in 3 dimensions looks within the healthy mouse coming from a healthy pregnancy. If you analyze the same thing in a mouse that is coming from a stressed mother, this is what we observe. We observe the hyperdensity and hyper-branching of neuron in those animals. And this was also correlated with hyperfunctionality and completely dysregulation of neurons that are responsible to sense mechanical stimulation. I'm not going to have to go really a lot in detail. I will focus the study on mast cell for the audience today, but there is in the article a really long demonstration in neurons as well. So we then studied the inflammatory component of the disorder. So we sorted CD45 positive immune cells from the skin, and we performed a single cell RNA-seq analysis if the pups were coming from a control or a stressed pregnancy. Again, here we found a normal percentage of immune cells, no difference between control and prenatal stress offsprings. However, what we found is that there is a huge amount of genes that were differentially regulated if we analyze skin mast cell, for example. We didn't find any impact on eosinophils, for example. So those genes were associated with, granule exocytosis and activation, energy metabolism. So we think, okay, those pups that are coming from a stressed pregnancy, they might have actually hyperactivated mast cells as well in the skin. So when you look at the face of those mast cells, in the absence of stimulation, we stand for intracellular granules using avidin here, and you can see that if the pups are coming from a stressed mother, their mast cell in the skin are already degranulated at birth. So do we need those mast cells to develop this phenotype of pediatric eczema? So we first use kid WSH mice that, they are deficient in stem cell factor receptor. Today they lack mast cell, but they also have other abnormalities. But those mice, they were fully protected from the development of the pathology. So we then obtained those mice from Axel Rohr's lab, the M75 Cre mice that are very specific for those connective tissue type mast cell, the one that express MRGPRB2 that come from the yolk sac, as I told you in the introduction. And when you deplete those mast cells that seed selectively the skin, you will find that those mice were also fully protected from the development of the pathology. So you need those mast cells of embryonic origin to develop the phenotype of pediatric eczema. So how does that work? So we then took a step back and we wanted to really look at within the fetus, what's going on when we stress the mother. So this is a cross-section of fetus that is coming from a controlled pregnancy. Mast cells are really coming early out of the yolk sac. They will seed the skin at the same time than macrophages. They will be very numerous. Nobody knows why we make actually mast cells so early in the body, but this is the case. And this is the mast cell, if they are coming from a PEPs that is in a controlled environment, a non-stressed mother, they are not degranulated. And again, if you look at those mast cells in the skin, just after the stress event of the mother, you start to see that they are completely degranulated in the skin. And these are the very same mast cells that will persist after birth in the skin of the offsprings. So to understand what was going on the transcriptional level, so we sorted those mast cells. So we used the stem cell factor receptor and the IES33 receptor ST2 to gate on fetal mast cell, because they, at this stage, they actually do not express the high affinity Ig receptor. And we found that those mast cells, where they are coming from a stressed pregnancy, the fetal mast cell, we found more than 1,000 genes that were differentially expressed. So they are very different than mast cells coming from a controlled environment. Again, these were genes associated with granule formation, remodeling, maturation, and activation as well. So what is dysregulated in the mother that actually would then imprint those mast cells in the fetuses? So for that, we took advantage of these very nice single cell RNA-seq data sets coming from the fetal skin. And we virtually isolated mast cell based on the carboxypeptidase 3 expression. And in the same times, we wanted to have a control population of immune cell that will also seed the skin and will be developing at the same stage, which are macrophages, thanks to CSF1R. And in the body, we have two main stress response systems. We have the HPA axis that will lead to cortisol in human or corticosterone synthesis in the mouse that will act through the NR3C1, the glucocorticoid receptor. And you will have the adrenergic response system with noradrenaline that will respond to the adrenergic beta receptor 2, for example. And what we found is that mast cells express very high level, much higher level than macrophages of the glucocorticoid receptor NR3C1, the fetal mast cell, while the macrophages are the only one that express the adrenergic beta receptor 2. So we think, okay, mast cell might be actually a target of glucocorticoid that would be secreted by the mother after the stress station. So when we analyze the amniotic fluid, we dose the level of corticosterone, and we indeed found a strong increase in corticosterone in the amniotic fluid of the PEPs after you stress the mother, meaning that when your mother is stressed, those babies, they start to bath, and they also swallow the amniotic fluid, in an environment that is highly enriched in corticosterone. So we then isolated ex vivo fetal skin mast cell, and we incubated them either with a control or directly with recombinant corticosterone. And indeed, those fetal mast cells, they are highly sensitive to corticosterone. They start to degranulate if you just put corticosterone on them. Then we did the same experiment, but putting those mast cells in culture with the amniotic fluid of a controlled pregnancy, so a non-stressed mouse. Nothing happens, but if you put them in culture with the amniotic fluid that is coming from a stressed mother, then those mast cells, they start to degranulate. So is there a way to inhibit this? So we use this molecule, the metirapone, which is a selective inhibitor of the enzyme responsible for corticosterone synthesis. So we develop a protocol in which we perform just a transient inhibition of the synthesis of corticosterone just before each stress event, such we can actually restore normal level in the peripheral blood of the mother, of the pregnant mother of corticosterone. This is what you see here. This is the blood. Controlled pregnancy, stressed pregnancy, you increase the corticosterone level, but when you pre-treat with metirapone, actually you normalize back to the basal level of corticosterone. Even if the mother is stressed, they have a level of corticosterone that is comparable to control mothers. And when we did that, we found that the PEPs were fully protected at birth. Again, no possibility of induction of pediatric eczema. Here is just to show you, it's a von Frey-Filaman assay that stands for mechanical pain sensation in the mouse, which was completely dysregulated. I didn't show you before in stressed mother, but if you pre-treat with metirapone, you restore also a normal mechanosensation, so a normal perception of mechanical sensitivity in the mouse. And then those mast cells, again, they adopt a normal phenotype. They are not degranulated anymore. They are not dysregulated anymore. So is it true as well in human? Just anticipating the question in the chat. we took advantage of basically human fetal skin mast cell from this nice paper. And also we isolated again virtually human macrophage and human fetal macrophage and human fetal mast cell that would also develop exactly in the same time. And we found that the level of expression of the glucocorticoid receptor and the adrenergic beta receptor 2 were comparable to what we found in the mouse, meaning that mast cells are the one that express the glucocorticoid receptor and macrophages are the one that express the adrenergic beta receptor 2. And atopic individual and stress is a long story. Atopy is often associated with hate and anxiety, stress-induced exacerbation of condition, and increased risk of eczema in offsprings. And we also know from the literature that newborns with a family history of atopy display an enhanced HPA axis response and response to corticosterone. So we took advantage of this very nice cohort named Healthy Early Life Moments in Singapore. It's in collaboration with Anand Andyapan, Jari Chan, and Florent Ginou from the A-Star and the KK Women and Children Hospitals. When they follow actually pregnant women during the pregnancy period, they characterize them into atopic or non-atopic women. And we had access to blood collection either during the gestational weeks or at delivery. Because delivery, we know that there is a boost of cortisol actually that enables the delivery that happens smoothly as a control. And so what we observe is that during the pregnancy period, if you are an atopic individual, you have almost twice more actually cortisol in the blood than if you are a non-atopic person. And then finally, during delivery, there is exactly the same level of cortisol. So we propose a revisited version of the etiology of very early pediatric eczema with remission that would be centered around in utero dysregulation of neuroimmune circuits. We think that during a chronic psychosocial stress, actually there is activation of the HPA axis in the mother that will lead to cortisol or corticosterone in the mouse in the amniotic fluid. Then this will completely dysregulate yolk sac-derived mast cells. We did and sensory neurons in the same times. We did a lot of fate mapping also. We show that those yolk sac-derived mast cells, they persist after birth, and you have a mixture of mechanical hyperreactivity that is driven by sensory neuron and a hyperreaction to inflammation that is driven by mast cell. We still don't know whether the two systems are connected. We have no evidence of connection. And then when those yolk sac-derived mast cells, we found that actually after 6 months in the mouse, which might happen much longer after years in humans, they are actually replaced by new mast cells that are still don't know whether they are coming from the bone marrow or from other progenitors, but those yolk sac-derived mast cells, they are replaced by mast cells that are not exposed to a stress environment anymore, that are completely normal, and then you switch back. Basically, you go in full remission of the pathology. So with this, I would like to thank all the members of my lab and of the GenoSkin team as well, and especially here Lilian, Nasser, Nadine Geziel, and former members Marie Taubert, who is now a PI in Lyon, and Nadine Serran, who is now a postdoc with Myriam Merad in the Mount Sinai, and all the collaboration. Also Philipp Starkl, which we collaborate, and all the sponsor and we have 2 postdoc positions open in the lab, so feel free to open. And thank you for your attention.
Thank you so much, Nico. Great presentation and great work and very exciting. We are almost out of time for your spot, but we take one question from Marcus Sperandio. Corticosteroid treatment is a common measure in pregnant women with threatened preterm birth to improve lung function in the premature infant. Did you check whether this treatment increases the risk of atopy in the offspring?
Yeah, it's a very good question. It's something that we are currently trying to address with the Singapore cohort as well. So there is a vulnerability period of during which those mast cells they develop. It could be that actually at the preterm birth, just before birth, actually those mast cells would be already developed and already imprinted. So this might not necessarily affect the behavior of the mast cell. However, something very interesting that we now investigate is the dermocorticoid treatment of pregnant mother. So this is very common. And we are now trying to see whether dermocorticoid treatment in mother would increase the risk of allergies in kids as well. So, but these are very good questions.
Thank you so much, Nico. There are a couple of more questions. Maybe you can answer them in the QA
Absolutely. I will.
chat. Thank you so much, and thank you for the questions. So we move on to the next speaker, who is Professor Jean Marshall. She is at the Department of Microbiology and Immunology in Dalhousie University, Halifax, Canada. She earned her PhD in immunology from the University of Manchester, UK. And I can mention that already then she started to work on mast cells and their function, which she has then continued to investigate these cells through her career. So after her PhD, she moved to McMaster University in Hamilton, Canada, for a postdoctoral training in the laboratory of John Bienenstock. And for the past 25 years or more, she has been at the Dalhousie University, where her lab has been recognized for pioneering work on mast cells, toll-like receptors, and antiviral immune responses. As an elected fellow of the Canadian Academy of Health Sciences, her current research focuses on mast cell-based therapeutics in viral infection and cancer. And Jean will then present today work entitled Mast Cells Both Enhance Early Effective Immunity and Promote Eosinophil Accumulation in a Model of Respiratory Syncytial Virus Infection. So please, Jean.
I'll move on, and thank you very much for the opportunity to present, and I'll just share my screen. Oop. I seem to... Can't see my... Oh, yes, there we go. Okay. So... Apologies, this is taking a little longer than it should. Okay, so I'm very pleased to have the opportunity to present today, and I'm going to be talking about some work we've been doing on respiratory syncytial virus infection. And this is work led by postdoc Rupa Habandi Nanjan Dapper, as well as other people from my lab, most notably Dr. Ian Heidel and Dr. Chris Liskey. And with trouble here. Okay, so mast cells are known to be important as sentinel cells, and particularly as sentinel cells in bacterial infection and parasites, but also in viral infection. And work going back from multiple labs over 30 years has shown that mast cells can respond to viral infections of various sorts, either by producing cytokines and chemokines, by degranulating, or by themselves becoming infected. And the roles of these cells in viral infection can include a prominent type 1 and type 3 interferon response, enhanced NK function, largely mediated by those interferons, effector cell recruitment due to downstream cytokine and chemokine production, and increased vascular permeability, particularly in the context of secondary infections. In addition, it has been shown in some circumstances that mast cells can act as a viral reservoir. However, the role of mast cells in vivo remains uncertain in a lot of situations and hasn't really been actively investigated in many viral models, particularly in viral models of respiratory viral infection. Oops. So we have a particular interest in RSV, or respiratory syncytial virus infection. This is a single-stranded RNA virus belonging to the Pneumoviridae family, and it's a major cause of hospitalizations amongst children, with approximately 100,000 deaths in children reported worldwide due to RSV, according to the World Health Organization. RSV is also responsible for a significant number of respiratory tract infections in an older age group, particularly those over 60 years of age. And severe disease is associated with eosinophilic bronchiolitis, and eosinophils in various models have been implicated in both tissue damage and in the effective host response against RSV. So some years ago, we began to look at RSV in terms of the ability of human mast cells to respond to RSV infection. And what we found was that while only a very small percentage of mast cells, in this case human cord blood-derived mast cells, become actively infected with RSV, the interaction between mast cells and RSV led to the production of both interferons and downstream chemokines such as CXCL10 and CCL4. And this we could see at both the protein and mRNA levels. However, mast cells didn't degranulate in response to RSV infection, and we found also that much of the downstream chemokine production was due to an autocrine action of interferons on mast cells. So more recently, we revisited the RSV model and found that in humans again, that human cord blood-derived mast cells, if they were treated with interleukin-5, and with elevated interleukin-5 being associated in some cases with infection, that if the mast cells were treated with interleukin-5, they didn't show an increased level of RSV infection, as shown by the gene N here on the left-hand side. However, they did show elevated levels of interferon response, particularly interferon alpha-2, interferon beta-1, and interferon lambda-1. So this suggested to us that IL-5, there might be a greater interaction between IL-5, potentially eosinophils, and human mast cells in this context. Now, this impact of IL-5, we've shown in published data that is due to increased ability of the mast cells to survive as a result of an EPAS-1 dependent anti-apoptotic mechanism. So what about the role of mast cells in vivo in response to RSV? So in this case, in order to do that, we looked at an appropriate mouse mast cell deficient model. In this case, we chose the Hello Kitty mouse developed by Dr. Galli and Dr. Tsai, which is not a c-Kit dependent model, but a c-Kit independent model of mast cell deficiency affecting all of the different types of mast cells, both those in the skin and in the airways. And we infected both mast cell deficient mice and their mast cell containing littermates with a mouse adapted RSV strain, and then examined viral load, the innate and adaptive cell populations in the lung, and inflammatory mediators in the airways, as well as airways histology, early post-infection, particularly focusing for the purposes of today on day 2 and day 7 post-infection. So what did we see? Well, the first thing we noted was that when mast cell deficient mice were infected with RSV, and we followed the mouse weight over several days from day 0 onwards, that the mouse cell deficient mice lost, as shown in the orange here, lost sufficiently more weight than the infected RSV wild-type mice that had mast cells. The next thing we looked at was the viral load, and we assessed this by drop digital PCR so that we could get an absolute value of the amount of virus in the lung tissue. And here is shown on a log scale, and you can see that the mast cell deficient HK mice, as shown in the orange, had a much greater viral load at day 2 post-infection than the wild-type mice at the same stage. So this indicated that the mice, if they lacked mast cells, were more badly affected by the RSV and were less able to eliminate the virus at this early stage in infection. So how was this occurring? So the next thing we did was to look at the inflammatory cells in the lung tissue at this early time point, day 2 post-infection. And what we noted was that while the numbers of neutrophils, dendritic cells, and TMB cells were not affected by the presence or absence of mast cells, the number of inflammatory monocytes was greatly increased in the Hello Kitty mast cell deficient mice, as shown in the orange, and the number of eosinophils was greatly decreased in the Hello Kitty mice, as shown in the orange. And this was clear both from the percentage of cells out of the CD45 positive cells and also at the level of the total number of cells in the airways. We also looked at day 7 post-infection, and by this time, most of the virus has already been cleared in this model, and there are no longer a significant difference in the amount of virus found in the lung in the between the HK mast cell deficient mice and the mast cell containing mice. And there are also no longer a difference in the number of inflammatory monocytes. However, the number of eosinophils was still in the lungs, was still decreased in the Hello Kitty mast cell deficient mice compared with the infected wild type mice. So this showed that there was a persistent change in the ability of the mast cell deficient mice to recruit eosinophils in response to viral infection, even at a stage where the viral loads were very similar at day 7. There was little impact of mast cells on the overall T cell responses to RSV in the lung, so we didn't see any changes in the expression of the major markers at day 7 post-infection. However, we haven't specifically examined the antigen-specific T cell responses in this model. So what could be driving these changes in inflammatory response and cell recruitment? We looked at a number of chemokines and cytokines in this model at day 2 post-infection in both the wild type mice and the Hello Kitty mast cell deficient animals. And as you can see, in general, most of the mediators we looked at were increased as a result of infection, as shown in the orange here compared with the blue for both wild type and Hello Kitty animals. So for CCL2, CCL5, IL-6 and IL-5, there were very similar responses in both mast cell deficient and mast cell containing animals. However, elevated CCL4, TNF, and CXCL10 responses were observed in the mast cell deficient mice following infection. And these elevated responses appeared to mirror the elevated levels of infection in the lung rather than the presence or absence of mast cells. To further evaluate what was really going on in the lung tissues, we completed an analysis of the lung histology with the help of our colleagues in pathology. And for this, we used an established scoring system and noted that the Hello Kitty mast cell deficient mice at either day 2 or day 7 post-infection showed a more severe histological damage score. And you can see this on the panel on the left-hand side at both day 2 and day 7 post-infection, that the wild-type mice clearly show impact of infection, but is not nearly as severe as that observed in the mast cell deficient animals. Well, no examination of mast cell deficiency in a model is sufficient unless you also do a mast cell reconstitution to show that the changes you're examining are actually due to mast cells and not to other cell types. And this is true whether you're using a c-Kit dependent or a c-Kit independent model of mast cell deficiency. So we performed experiments where we took bone marrow cells from wild-type mice, we cultured them, and then derived bone marrow-derived mast cells. We then reconstituted the mast cell deficient mice intravenously and examined them in concert with age-match mast cell deficient and mast cell containing animals. And what we saw, as you can see from these graphs, is that compared with when we use wild-type mice, this is virus levels of virus infection are shown in panel A here. You can see that the wild-type mice shown in blue showed a significantly reduced level of infection at day 2 compared with the Hello Kitty mast cell deficient mice. But if we reconstitute, the levels of infection return to those very similar to those observed in the wild-type mice. Similarly, if we look at weight loss, the wild-type mice showed less weight loss than the mast cell deficient mice, and this less weight loss was recovered when the Hello Kitty mice were reconstituted with mast cells. Similarly, mast cell reconstituting normalized the inflammatory monocyte response and also normalized the eosinophil response at day 2 post-infection. So this very much confirmed that not only that the mast cell deficient mice had a worse response to the RSV infection, but also that this could be fully reconstituted, fully recovered by reconstituting the mice with mast cells. So we then considered the role of eosinophils in this system. So the eosinophil story with respect to RSV is complicated, and I'm not going to do it justice here, but I'm just going to give a... So if it's known that there is an elevated lung eosinophil response, and we observed this too, that following infection in the presence of mast cells. So we wanted to know if that was responsible for the reduced infection and weight loss, or whether the eosinophil response was an indication that mast cells were present, but not actually responsible for that early change in host defense. And it has been shown that eosinophils have a protective role in RSV infection, but this is based mainly on work on IL-5 blockade and IL-5 using IL-5 blockade or IL-5 transgenics, and particularly at day 3 and beyond in infection, so not at this very earliest stage in infection. But because of our recent work, we also know that IL-5 has other impacts in the infection model, particularly enhancing mast cell interferon responses. So it makes the interpretation of those earlier studies a little bit more complex. So in order to address that, we looked at the early response to RSV in DBL Gata eosinophil deficient BALB/c mice compared with control wild-type BALB/c mice. So these mice are both on a different background from the mice we were looking at earlier. And the bottom line is that eosinophil deficient mice did not demonstrate any impaired early response to RSV at day 2 post-infection. So we didn't see differences in weight loss, we didn't see significant differences in viral load, and we didn't see differences in inflammatory monocyte recruitment at this very early stage at day 2 in these eosinophil deficient animals. So in summary, mast cells are beneficial in controlling early RSV infection in this model. So in the presence of mast cells, including following mast cell reconstitution, we observed a reduced initial day 2 RSV viral load, an increased eosinophil recruitment at both day 2 and day 7, a reduced lung pathology at day 2 and day 7, reduced inflammatory mediator responses, and reduced inflammatory monocyte recruitment. So really very clearly demonstrating that mast cells at early stages of infection are likely to be very protective in this model. We also noted that eosinophil recruitment is not linked to increased pathology. So if you take the mast cells out of the equation, the recruitment of eosinophils is not associated with worse pathology. In the presence of mast cells, there are lots of eosinophils recruited, but there's an improved pathology. We also showed that eosinophil deficient mice do not share the reduced ability to combat early day 2 infection observed in mast cell deficient mice, really pointing the finger of mast cells being particularly important at the very early stages of infection. So what are the take-home messages? First, this data suggests that mast cells are critical for effective early defense against RSV infection. Obviously only in mice, but it would be interesting to know more and use different models to find that out. The mast cell-mediated recruitment of eosinophils following infection is not associated with more severe lung pathology, suggesting that targeting that pathology may be more effective targeting cells other than eosinophils, and that very early host defense against RSV does not appear to be eosinophil dependent. So obviously there's a lot of work to do in this field, and this is only one mouse model, but we think that these findings are of interest and represent a clear indication of a in vivo role for mast cells in early host defense against viral infection. Finally, I'd like to acknowledge the people who are most important in doing this work, particularly Dr. Rupa Nanjundappa, Dr. Ian Heidel, and Dr. Chris Liskey. The work on IL-5 and human mast cells was done by excellent postdoc, Dr. Jasmine Barra. And I'd like to thank my funders, particularly the Canadian Institutes of Health Research, for this work. And thank you for the opportunity to present.
Thank you so much, Jean, and also then connecting the mast cells and the eosinophils in the last presentation of this webinar. So we have time for a few questions. I'll start here with one from an anonymous attendee. Can RSV reach the brain? What is the role of mast cell-derived serotonin in RSV reaching the brain?
Can RSV reach the brain? I don't know. It's a short answer. I mean, we have used some other models of viral infection where we have seen brain infection, and certainly the intranasal route does provide that opportunity, but we don't see any signs of morbidity that would be associated with brain infection in this model.
And Jenny Hallgren Martinsson writes, very interesting. Did you check for influx of mast cell progenitors in the lung at day 2 after RSV in wild types? Do you think the response to RSV is due to the very few resident CTMCs?
As you know, in these, as many people in this audience will know, in the black 6 mice, there's actually remarkably few mast cells in the lung parenchyma. So our current working theory is that it's actually the mast cells in the upper airways that may be more important in this very early response, such that potentially even the effective viral load that reaches the lower airways is reduced due to the presence of mast cells. But we don't have good data to support that yet. Another possibility is that the pleural mast cells play a role, but given that this is day 2, my money is on the upper airways at this point.
Okay, thank you so much, Jean. And you have a couple of more questions to answer in the QA, if you can do that. And I will hand over to Allison Fry to summarize this great webinar. Thank you all.
Well, thank you so very much. So I'd like to thank the presenters for this fabulous talk this morning. There were 3 really amazing presentations. So thank you all so much. I'd really like to thank EMBRN and especially Dr. Nilsson for co-hosting with us this morning. And finally, all of you will be receiving a post-webinar evaluation. Please look for this in your email and answer this if possible. We do actually look at the evaluations and use them to plan future seminars going forward. And finally, I'd just like to remind everybody that the entire seminar will be available online on the IES website. So thank you all so very much for an amazing session. And I look forward to seeing everybody at some point in the future. Thank you.